Hi. So now we're going to start systematically going through the micronutrients. So we're going to have two chapters on carbohydrates. And this first chapter, chapter 8, is labeled sugar. And it's not just going to be the sugar, the table sugar that we know, but all of the sugar carbohydrates, which are um, the simple sugars, so mono and disaccharides, which we'll learn about this week. So the way we're going to do this chapter, I'm not going to jump around as much as I have some of the other chapters. We're just going to start at the beginning and go through, I think it's page 222 is where we'll stop. Um, and just straight through the whole first half of the chapter approximately. We'll be covering the chemistry of sugars, and then start talking about where we get a lot of the sugars. And then next week we'll cover more of the functionality of sugars as well as nutrition. So, okay. So carbohydrates in general, this overview of carbohydrates, not just the sugars that we're talking about. Carbohydrates are 50% of Earth's biomass. And by biomass, I am including all of the living organisms plus their waste products. So another way that we could refer to that is organic matter. Okay. Um, if we just narrow it down to plants, carbohydrates are 75% of the dry mass of all plants. And as you'd expect, because it is such a big part of the biomass of the earth, it's going to be a big part of what we consume. And so the dietary guidelines for Americans, so government recommendations for nutrition, recommend that your total calories consist of about 50% of your total daily calories consisting of carbohydrates. And that equates to about 275 carbohydrate grams per day. This figure here is showing carbohydrate consumption over time. So you can see carbohydrates have increased a little bit. And actually this is food disappearance. So it's not consumption. So this is from the Food and Agriculture Organization of the United Nations. And one of the ways that they kind of indirectly measure food consumption is food disappearance. How much food is sold? Okay, because that's easier to find out than surveying everybody in a country. How much exactly did you eat? And so they can measure more economically, like using the economics, how much food has been sold, and what foods have been sold, and then kind of take a guess at where then the consumption is of those foods. So you see that the kilocalories here is well over 3,000. The average, or Americans are not consuming on average over 3,000 kilocalories per day. Um, and so this is a little bit off, but the proportions are probably pretty accurate. So you can see carbohydrates, they've increased a little bit since the 1960s as a percent of our diet, but fat has increased quite a bit more, and protein has remained pretty stable over time. So carbohydrates are composed, this is important, of carbon, oxygen, and hydrogen, and hence the name carbon, hydrate is water, right? Hydrogen or hydrate means water, and so hydrogen and oxygen are what water is composed of. So the empirical formula of carbohydrates, what most carbohydrates fit into, is going to be C with some number of carbons plus, and this should be subscript, but H2O and some number of that. And in general, that that proportion where there's always like an H2O and there's always going to be 2 hydrogen for every oxygen. In general, that works. There are some exceptions, and we will get to that in a little bit. Also, this formula is different than what we learned in chemistry, if you were in my chemistry class last year, and there is a reason to that. What we learned in chemistry is not incorrect, but we will get to that in a little bit also. So, carbohydrates are plants' means of storing energy. And here is photosynthesis. So if you remember from biology, plants will convert, or plants get light, and one of the outputs of photosynthesis in this overall reaction is glucose. And plants will convert that glucose that's produced during photosynthesis into whatever form of sugar, starch, or fiber that they need at that time. So fiber is used for structure, sugar and starches are going to be used for energy storage. So the types of carbohydrates. These types that I'm telling you are slightly different from what's in the book, and I want you to know the types that I'm telling you and not what the book is telling you. There is a review question that asks you the types, and I'm expecting to see listed what I'm telling you and not what the book is telling you. Okay, so types include sugars. These include these also can be called simple carbohydrates, which is another, or simple sugars, and saccharides. Anytime I have something red here means that I'm telling you something that is different from the book and you should be paying attention, okay? So another name for sugars is simple carbohydrates, okay? And I'm including all of the sugars here. I believe that the book only calls monosaccharides simple carbohydrates, but the real definition of simple carbohydrates is that it's mono and disaccharides, not just monosaccharides. So definition of simple carbohydrates that I'm explaining is different from the book. All sugars are simple carbohydrates. Okay, so within sugars, this is just 1 or 2 sugars that are bonded together, or there's 1 sugar or 2 that are bonded together. Okay, and then the next type is complex carbohydrates. So complex carbohydrates, this actually should be read here, okay? So complex carbohydrates, this is what's different from the book. The book puts it into, let me make sure I'm telling you exactly the right thing from the book. Not in the right section. So yeah, so the book tells you the 3 categories are sugar, starches, and fiber. Okay, so I'm telling you that instead of starches, you're going to use complex carbohydrates. Okay, so these are starches and other carbohydrates that have 3 or more sugars bonded together, as long as they are not fiber. Okay, so fiber is specifically, fiber is also a complex carbohydrate, but it's specifically non-digestible. Okay, and so down here, these are the chemical structures of many of these molecules. So here are several single sugars. When we have 2 sugars bonded together, they're called disaccharides, and then amylose is one of our complex carbohydrates. It's also known as starch. Cellulose is fiber, and beta-glucan would be another complex carbohydrate. And so it's 3 or more bonded together in these cases. Okay, so before we get really into the chemistry of sugar specifically, we need to do a little bit of backing up and talk about some general chemistry, because you need to understand this before we get into sugar. Okay, so we have talked before about hydroxide ions. Okay, so a hydroxide ion has an oxygen and a hydrogen, and because that oxygen really wants to have something else that it's bonded to, it actually still has a negative charge. Okay, that hydrogen bound to the oxygen and took away one of its negative charges, but it still has a net -1 charge. Okay. A hydroxyl group then is when that oxygen that is bound to a hydrogen, so it's a hydroxide ion, binds to something else. Okay. When you have chemical figures, an R, it simply indicates something else. Okay. It can be anything that goes in that R position as long as it chemically works. Okay. But all that it's showing you is this is the group that we're talking about. This is the hydroxide group. And if it's bound to something, it is a hydroxyl group and not a hydroxide ion anymore. So, for example, in this chemical structure here, you have the OH, right? And it's just showing all the bonds here. And it is bound to a carbon, and that carbon is bound to some other things too. It doesn't matter, okay? But this is the red here is the hydroxyl group. Okay, the reason this is important before we get started talking about sugars is that sugar has a lot of hydroxyl groups. Okay, so if you look at this figure here of glucose, you see 1, 2, 3, 4, 5 different hydroxyl groups. This OH is actually bound to the carbon. If we were to draw it out, there'd be a line here, a line here, and a line here. And 2 of the lines would be hydrogen, and 1 would be the hydroxyl group there. Okay, so 1, 2, 3, 4, 5, sorry, 1, 2, 3, 4, 5 different hydroxyl groups that are in a single glucose molecule. So they're pretty important to what makes a sugar a sugar. Now, monosaccharide. So mono means one, saccharide means sugar. So monosaccharides are sugars that contain just one basic molecule. Okay, one basic sugar molecule. Okay, the formula, the empirical formula for a monosaccharide is going to be CH2O and N. Okay, so it's a 1-to-1 ratio of the carbon and the water. Now, there are 2 exceptions to this rule, and that's why the book says, like, we used to think that the formulas for carbohydrates were CXH2OY, but we know that's not the case anymore because there are 2 exceptions, and those are a molecule called rhamnose, okay, and it has 1 less oxygen than it would if it were following this formula, and deoxyribose, which makes up DNA, as part of DNA. It also has 1 less oxygen atom. And interestingly, or if it helps you remember this, deoxy means that that it's missing an oxygen atom. Okay, that's what the deoxy in front of ribose means there. So the monosaccharides include glucose. In the food industry, it's often called dextrose. So if you see it as an ingredient in your food, it will be called dextrose and not glucose. So glucose can be naturally found as a monosaccharide in honey, in dried fruits. We don't find it so much in fresh fruits, and we'll get into why. Grapes and corn, and pretty a pretty small amount in nature, because most of the time our sugars are found as disaccharides or polysaccharides, complex carbohydrates. Okay, But glucose is the most common monosaccharide in nature. And like I said, it's more commonly found as a di or polysaccharide. Glucose is one of our main sources of fuel of humans and animals. Okay, we find it in our blood. It is being constantly transported as a source of energy to different parts of our body. Okay, the second major monosaccharide is fructose, and we find that in fruits and honey. And finally, we have galactose. It's a third one that we'll talk about, and that is found mostly in milk. It can be found combined with other sugars in other parts of nature. It can also be. It says, I think it says in the book that it's pretty much only found in milk, but there are certain fruits, especially, and maybe one or two vegetables that galactose can be found in very small amounts. Um, yeah, and like I said, it's found more in more than just milk, just not as a monosaccharide. And even in milk, it's predominantly part of a disaccharide. Galactose is not very stable by itself, so it very easily forms bonds and becomes part of a disaccharide. Okay, so what does the structure of these sugars look like? Okay, so if we were to draw it with, if you were in chemistry, some of like the figures that we've used before with chemistry, it would look kind of like this. And people used to think that sugars were linear like this, right? You have all these carbons, and then you have the hydroxyl group sticking out on different sides of it. This is the carbonyl group. And they really kind of found out, wait, that's not usually the case. It can exist in this form, but in aqueous solutions, which is most solutions that we're finding these sugars in, because we're finding them in biological material, which has water associated with it, they will be in a ring-shaped structure instead. Okay, and so what happens here is it closes in on itself, and this oxygen here, instead of having a double bond, it gives like one of those bonds to this carbon at the end, okay? Or at the beginning, I should say. Okay, so this oxygen binds to the first carbon, and that's what we're seeing here. This is the first carbon here, and the oxygen that was at the end there is binding to that first carbon. Okay, so glucose and galactose, notice they look different than fructose. So the glucose and galactose have what we call a 6-member ring. If you're drawing it out like this, it is a hexagon. Okay, hexagon. And fructose has a 5-member ring, so it looks like a pentagon. Okay, so these 6-member ring sugars, we call them hexoses. Hexagon, hexose. Okay, the 5-member rings we call pentoses. So pentagon, pentose. If it helps you to remember which is which, fructose, F comes before G, right? So F has 5 and the G's have 6. Okay. Note, also want to point out some something that is helpful to know, especially if you go on into college and have to take a chemistry class, is how some of this is drawn. So this figure from before, I showed you all the carbons, right? But on those carbons are not written out. And the reason for that is, number one, like if you're writing it out, it saves time, but it also saves clutter. This clutter of the image, okay? So anytime that you see lines that there's a bend in them, but there's no actual atom element written, you assume it's carbon. Okay, and this is just because all these organic materials, organic molecules have lots of carbon in them, and so it saves on clutter and time to just say, okay, if there is a bend, an angle in this molecule, and there's nothing written, it's carbon. Okay. Um, also, so on this one, okay, you'll notice that there's also a hydrogen that is in these, but not here written, and it actually is there. It's another assumption that just, I guess, chemists get lazy or efficient, however you want to think about it, and they don't include all of the hydrogens that are bonded to carbons, because you can also pretty much assume carbon needs to have four bonds. Okay, it wants to have 4 bonds. And so if, let's say I'm looking at this carbon here, okay, so I see one of the bonds is this hydroxyl group. Another of the bonds is to this other carbon here. So that's 2. The third one is to this carbon. So I have 3 bonds. Any bonds that aren't explicitly spelled out, there's a hydrogen on there. And so even though it doesn't show that there's a hydrogen like it does on these, it is there. Okay, and the only explanation I can think of why they didn't put the hydrogens on this one, even though they did this one, is because it's a pentose, so there's a little bit less space to put all of the bonds in the structure that they've drawn out. Okay, so not super important for what we're doing in this class, but something to know for future chemistry stuff. Okay, so glucose has many forms, okay? And uh, uh, so there's two main forms, all right? There's D-glucose and L-glucose, okay? And what is different about these is that, okay, so when we see a D before glucose, it means right-handed, and L means left-handed. And what is going on here is actually just one spot that's different. It's where this CH2OH group is located on the molecule. So for the D-glucose, it is on the top side, and for the L-glucose, it is on the bottom side. The only reason I'm really telling you this about D versus L is because in the book, it starts talking about alpha and beta glucose, and all of a sudden it adds this D. It's alpha D glucose, beta D glucose, and doesn't tell you anything about where that D came from. So I'm just letting you know that's where it comes from, is because there are different forms of glucose, and the D means it's the right-handed form. Okay, now this L form is actually not found in nature. It can be synthesized, but it's not going to be in any of our food. So X out here, everything we do from this point forward is using D-glucose. Now, alpha and beta glucose, which is where we're ultimately going with this, as you can see from the title. So within D-glucose, there are 2 configurations of interest, okay? And that is alpha and beta glucose. And their only difference is how this hydroxyl group is located, or is rotated, you could even say. So on the alpha D-glucose, the hydroxyl group goes down at this point, and on the beta D-glucose, it goes up. Now, this small difference completely changes how they react to form bigger structures, and how the human body digests them. We are able to easily digest alpha glucose. However, we don't easily digest beta glucose. It just doesn't fit correctly within our digestion enzymes. Um, and so alpha glucose is our main energy source, and beta glucose is actually the main component of fiber. And part of that even is how you'll see when we get to complex carbohydrates, how these combine together to make a longer chain carbohydrate. They look very different when they combine too, because of where this hydroxyl group is situated. So everything that we've talked about so far is, I told you something wrong. Okay, let's go back for a second and change something that I told you wrong. I called this a pentose. This is actually not a pentose, so don't get confused with that. It is a 5-member ring, but they are all what we call hexoses because they actually all have 6 carbons. I'll show you that, even though it doesn't look like it at first glance here. So on the glucose, you have 1, 2, 3, 4. Hold on. Where am I missing? 1, 2, 3, 4, 5, 6. Okay, there's one here too. Okay, and then so the same thing here. They're in the same locations. On the fructose, notice right here, there is a carbon that is just hanging out off to the side. You still have the hydroxyl group, but instead of a hydrogen bonded there, you have a carbon group here. Okay, so there's 1, 2, 3, 4, 5, 6. Okay, so all of these are hexoses. Now that I've fixed that, okay, so everything that we've talked about so far is a hexose because it has 6 carbons. And so the formula for all of the hexoses, so whether it is glucose or fructose or galactose, they actually all have the same chemical formula. The chemical formula doesn't tell us everything that we need to know about a molecule. How they are actually physically are put together, their actual like 3D shape is really important to what the molecule is. So all of the hexoses that we've talked about, glucose, galactose, and fructose, are all hexoses with the chemical formula C6H12O6. All hexoses have this formula because they're following that CH2O empirical formula, except rhamnose, which I mentioned earlier. Okay, there is also a group of monosaccharides called pentoses, and these pentoses are going to still following that CH2O and then with an N on the outside of the parentheses formula, C5H10O5. Okay, so there's twice as many hydrogen as there are carbon and oxygen, except deoxyribose is a pentose that does not follow that. Both of these have 1 fewer oxygen in them than this formula. So this one has 5 oxygen and deoxyribose would have 4. Okay, so included in the pentose is ribose, and ribose is really important because it is part of RNA. And it's one of the building blocks of RNA. It helps build this strand that is the backbone of RNA. Now, deoxyribose, which is that exception here, okay, that has an oxygen taken out of it, and you can see that oxygen taken out of it right here. Ribose has it there, and deoxyribose does not have it, and there is a hydrogen there. So you have a line here, and there's nothing at the end of it. There's a hydrogen there. So deoxyribose is our main component of and the backbone of DNA. Okay, so now we'll talk about disaccharides, which are two monosaccharides that are bonded together. Okay, we don't, like I said before, we don't find a lot of monosaccharides in nature. More often we find these simple sugars as disaccharides or more complex as polysaccharides. Okay, so sucrose is the first one that probably used the most as an ingredient in food science. It is what we commonly refer to as sugar, right? So if I'm just talking about add some sugar, you're going to add sucrose unless I've specified. What you see at the store labeled sugar, this is sucrose. So sucrose is one molecule of glucose and one molecule of fructose that have bonded together. Lactose is going to be a molecule of glucose, which is here, and a molecule of galactose bonded together. So lactose is found in milk. That is the main place we're going to find it, almost nowhere else. Okay, it contributes to the white color of milk as well. Okay, and then maltose is going to be two glucose molecules that are bonded together, and it is the least sweet between these disaccharides. So if you're just taking a taste, it will not taste as sweet as the other two on a mole per mole basis. Okay, so you probably didn't notice this in the previous slide, but I'm going to point it out now, is that when these two molecules, whatever monosaccharides we're talking about here, we're doing two glucose and making a mannose. When these two molecules bond together, for that reaction to occur, we actually have to lose a water molecule. Okay, and so you notice like there's two hydroxyl groups here, right? And where they were bonding, and now there's only one oxygen, right? And so one hydroxyl group and one of the hydrogens had to, or one hydroxyl group and one of the hydrogens had to be removed from between these two molecules and to make this disaccharide. Okay, so the creation of a disaccharide is a dehydration reaction. Okay, now conversely, if water is added to these molecules, a hydrolysis reaction can occur. So hydrolysis means hydro, water, lysis, break apart. Okay, so you're breaking apart with water. How you can remember that term, right? And lysis is a very common biological term. Okay, now, usually can't just add water and it breaks apart. That's not quite how it works. And this next slide, we'll talk about that. So 3 conditions can trigger this hydrolysis. 1, an enzyme sets off the reaction. 2, the addition of an acid. Or 3, the addition of heat. So what I'm showing you here is, let's see, I mean, this would be just like a generic Erlenmeyer. Hey, let's add an acid. Here is an enzyme, all right? So you have the enzyme here, and the sugar fits just perfectly into that enzyme. It has to be a specific disaccharide or the enzyme is not going to fit perfectly with that sugar. And once it fits perfectly, it's like, okay, yes, let me help you do this. And water is added and the enzyme helps split apart the glucose and the fructose in this case to create 2 monosaccharides. So this would be a sucrose molecule. Now, another way you can do this at home with acid and heat. This image here is showing the person is boiling sugar water, so sucrose and water, and they add some kind of acid, often cream of tartar as an acid, and as the temperature goes up, we get it to a certain temperature, those molecules will break apart into glucose and fructose, and you have what is called invert sugar, which the two sugars separated is sweeter than the original sucrose solution that it was composed of, yet you still have the same number of sugar calories that are there. Okay, so enzymes, just getting a little bit more into that. So for each disaccharide, there is a specific enzyme in our bodies that digests it, splitting it apart into two monosaccharides that can be more easily used by our bodies. All of these enzymes are found in the small intestine. So sucrose, to digest sucrose and hydrolyze sucrose. So enzyme, we're talking about enzyme hydrolysis here, just continuing from what we talked about on the previous slide. Sucrase is the enzyme that is used. Another word for that enzyme, it's still the same enzyme, is invertase. And that is more of like a food science term, and it does the same thing, just splits apart the sucrose into glucose and fructose. Okay, so lactose, to digest lactose, we require the enzyme lactase, and maltose would be maltase. All right, it's really easy to remember. You just, instead of ose, it's ace, to know what the enzyme is that does the hydrolysis of those sugars. Now, on the invertase, so the reason it's called invertase is because, as I kind of mentioned in the previous slide, the fructose glucose mixture that is created when you use sucrase and split those up here is sometimes called invert sugar. And from the previous slide, you can create that on the stove, and it's sweeter than sucrose. So about 1.3 times sweeter than the same amount of sucrose. It also retains moisture better and crystallizes less easily than sucrose, so it has some functional benefits besides sweetness as well for the food scientist. Okay, so alcohols. An alcohol is any organic compound whose molecule contains one or more hydroxyl groups attached to a carbon atom. And so you're like, what? We have lots of hydroxyl groups in sugars. So sugars are alcohols? No. So sugars contain multiple hydroxyl groups, but they also contain a carbonyl group. And so, oh, it doesn't show it well on here. You remember from that earlier slide, I'm just gonna go back several slides here. Fine. Whoops, went too far. Okay. Okay, here. Okay, we had the carbon and a double bond with oxygen. Okay, and that bond changes a little bit when it goes into that ring structure, but it's still pretty much there. Okay, it's still considered a carbonyl group. So now go back. Okay. So if a molecule contains a carbonyl group, it is called either an aldehyde or a ketone. So And whether it's called an aldehyde or a ketone depends on where the carbonyl is. And due to chemistry naming conventions, you call the whole molecule based on which group has a higher priority. So carbonyl groups are a higher priority molecule. They affect more of the functionality of a molecule than the hydroxyl groups. Okay. And so even though we have lots of hydroxyl groups, because we have a carbonyl group in our sugars, they're not called alcohols unless we change something. Okay. So sugar alcohols have this carbonyl group replaced with another hydroxyl group. Okay. So up here would be where that carbonyl group is normally, and it's replaced with another hydroxyl group. Okay, so now we have nothing but hydroxyl groups as our little special groups attached to what was the sugar. Okay. These sugar alcohols can be found naturally in different fruits or vegetables. It can also be manufactured, usually using a biological process. Fermented with yeast or using some other microorganism that naturally creates sugar alcohols. And they have a structure that, and they're not alcohols either in a way. Like they're alcohols by naming conventions, but they don't behave like other alcohols, like ethanol or methanol or isopropyl alcohol or anything like that. But because their structure is similar enough to sugar to give a sweet taste, because I mean, if I just put this on the screen, you not being super familiar with sugars, you'd be like, oh, that's a sugar. It has all those hydroxyl groups, right? And your tongue, your taste buds think the same thing, right? So it has a structure similar enough to sugar to give that sweet taste, but with that functional group changed, now it doesn't fit in our enzymes like it did before, and it doesn't fit, and so it can't be metabolized like sugars can. Our bodies do not recognize it. And so it's not digestible. And because of that lack of digestibility, these sugar alcohols are fewer or no calories, which can be a great thing for people who are trying to avoid sugar. Now, on the bad side of that, their lack of digestibility can cause problems in our digestive system. It just goes right through, like other things. And so, like other things that just go right through our digestive system and aren't digestible, it does have the potential to cause bloating and potentially diarrhea if you consume too much of the sugar alcohol. So, consume in moderation. So now, sources of sugar. Sugar cane. So the process for the production of sugar from sugarcane. You have cane stalks that are harvested, taken to the mill. The stalks are washed, cut, and the juice is pressed. The juice is clarified. So like they try to get different materials out to make it look clear. Clarified means to be made to look clear. Concentrated, and then crystals are formed out of it. The crystals are spun in a centrifuge because those crystals are still sitting in a wet solution. And so that they're spun in a centrifuge to remove the liquid, and that liquid is called molasses, and this is different from what the book says. So pay attention to this definition, that molasses is a thick, dark syrup left over after removing the sugar crystals. Then the raw sugar is transported to a cane sugar refinery. So just to show you those steps, there's the sugar cane in the field. This is what it looks like close up, cut. Here they are cutting it, and they cut it just very finely, and so that the juice can be pressed out. Here they are pressing out the juice, and you can see it coming out. Here they are clarifying it, so they'll add different agents that bring different parts of the juice that is not sugar, either to the bottom to be use gravity to get it out, or we'll bring it to the top. Those are called flocculants, to make the stuff float and they can skim it off. So this is clarifying. So this is more clear than this, right? And so this process here, I think they add lye at this step, gets a lot of this stuff, this gunk out with gravity. Okay. Here, I think they are boiling and dehydrating it a little bit, if I recall. Here they've added the flocculant, and you can see there's these skimmers here, and they're just like the excess stuff. They actually call it scum at the top, and it pushes it off and over, like off the edge of the tank, and out of the tank, to get rid of those materials. Then they're going to actually add some sugar to the sugar. So this is sugar crystals that are in ethanol, I think. And so they add that, and those are seeds. And if you add crystals to sugar that has not crystallized, it will start to crystallize, attach itself to the crystals and form bigger crystals. So that's what's happening there. Then they put it in the centrifuge here. So the solution is going down here, and on the inside there's this tube, and it has lots of small holes, and the holes are smaller than the size of the crystals that have been formed, but big enough to let all the moisture out. And so they're going to spin this, and centripetal force is going to cause the liquid, which you can see kind of here. These are stills from a video. So this is the molasses coming out, just getting spun out of the centrifuge. And what is left is the drier raw sugar. Okay, and so that is what that raw sugar looks like. So then it's going to go to a refinery where it's going to be further processed. It's going to be washed, decolorized, recrystallized, packaged, and shipped out. Okay, so this is images of that. It's being washed. Then they add a decolorizing agent, and you can't see a lot of that because they have these big tanks and it's all hidden inside there. Here, I think they're drying it. Yeah, and there's like a tumbler that they're drying it out. And once it's dry, they put it into the package. So sources, sugarcane products include raw cane sugar. So that would be at that step before it goes to the refinery. You have that raw sugar. So that's what this like turbinado cane sugar is, often sold as sugar in the raw. So just less processed. Okay, that molasses that comes out, there's actually several steps that molasses can be created in, and it makes different grades of molasses with different sweetness and bitterness to them. Brown sugar. So typically brown sugar these days, it's really just white sugar that they've added some of the molasses back to. So fun fact, if you don't have any brown sugar, but you do have white sugar and molasses, you can make your own brown sugar. Just add a little bit of molasses to your white sugar, and it's exactly the same thing. So that molasses gives brown sugar a damp texture. And so if that moisture is lost, then brown sugar will harden. So if you ever had hard brown sugar in your pantry, that's because it has lost moisture. You can add back that moisture or keep it from losing moisture by adding something that has moisture but less sugar than sugar. So slice of bread, a cookie even probably work. Slice of bread would be easier. An apple slice, or even marshmallows. Some people will just put marshmallows in with their brown sugar, and it keeps it nice and moist and not hardened up. Okay, so then granulated sugar is the next one. Why is it not going down? Why is my thing not working? Okay, granulated sugar is the next one, and that is fully processed sugar with all the colors and minerals and everything else except the sucrose removed. And then finally, powdered sugar is granulated sugar that has been ground down to a finer powder, and most of the confectioner sugar that we see in the store is going to be 10x if you ever look for it. And that just tells you how much it has been uh, powdered down. So the finer it is, the higher the X number is. But you can find smaller X numbers, more of a specialty thing or specifically for food manufacturing. Um, and yeah, just not used by home bakers. So. Okay, so sugar beets is another source of sugar. And sugar beets use the process to extract sugar out of sugar beets is actually pretty similar to cane sugar, so I'm not going to go through the whole thing, except beets grow underground, right? So have to be dug up. And because they are completely dug up, they have to be replanted every year. Cane sugar is like a grass, and so they cut it and it comes back up, cut it and they come back up, comes back up. So they don't have to replant it every year. They just harvest it and fertilize it and take care of it. Beet sugar is much cheaper than cane sugar, even though they have to plant it every year. So just kind of FYI, if a label doesn't tell you that it is pure cane sugar, most likely you are eating beet sugar. So like I looked up these two. Obviously this one says cane sugar on the label, so it's cane sugar. But on this one, if you look in the ingredients, all it says is sugar. And Great Value, it's Walmart brand, it's cheaper, right? And so most likely this Great Value brand granulated sugar is beet sugar. Now, chemically, absolutely no difference. It's just sucrose. Okay, maple syrup. So maple syrup is the concentrated sap of sugar maple tree. So it's a specific type of maple tree that creates maple syrup. And they just like jam this thing with a hole into the tree and sap comes out into a bucket. They do cover the bucket, but this image is just to show you the sap in the bucket. And it's watery, all right? It's not like hardened tree sap that you might have seen on trees before. And so they then pour this into a vat, and they're basically going to evaporate off most of the water. And once they've done that and they've concentrated that sap, it is now maple syrup. Corn syrup. So corn syrup, they actually start with corn starch and they hydrolyze it. So they are breaking apart that starch molecule into glucose molecules using acid and heat and or enzymes. So high fructose corn syrup also uses enzymes to convert some of that glucose to fructose. And it is a very high concentration of fructose in the high fructose corn syrup, they will also use something called liquid chromatography, which is more advanced scientific technique to have even more fructose in there. So sucrose, if you remember, is 50% 50% fructose, 50% glucose. And so high fructose corn syrup is can have different amounts of fructose. So not all high fructose corn syrups are the same. And different types of high fructose corn syrup are used for different food applications. So the HFCS 70% fructose is used in filling jellies, like filled donuts, right? 55 is used in soft drinks, and 42 is used in lots of other things. Now, 42, that's 42% fructose. and 50% is what is in sucrose. So why would we use that? The HFCS 42, it is less sweet than sucrose because there is less fructose, but it is cheaper to use than fructose. So that is why that is used. Okay, so sometimes it's not for a specific sweetness benefit, but for cost benefit that the high fructose corn syrup is used. And it may have actually less fructose than sugar. So that's something to know too. Just FYI here, so this Karo, this dark corn syrup, it actually has a little bit of molasses combined into it to make it dark. That's why it's dark. Okay, sorghum is another source of sugar. It's made from the canes of sorghum plants. Okay, so sorghum, the seeds can be used for, the grains can be used for one thing, and the canes can be used for this syrup. It sounds a little bit like sugar cane, but it's not quite the same. There's no sugar crystallization in this process. It's just a syrup that's formed when the excess water is steamed off, kind of more similar to maple syrup production, although it's not sap. It's still the juice from the cane, but so it's kind of like a combination. It is thinner than molasses, and it has a little bit of a more sour taste than molasses. And then finally, honey. Honey, so bees get nectar from flowers. Nectar has complex carbohydrates, and it's 80% water. The bees convert it to simple sugars using enzymes, including invertase and amylase in their salivary glands. So they actually chew it up, spit it out, chew it up, spit it out. And that is how honey is made. So you're eating bee spit if you like honey. I do, but so the invertase or sucrase, right, breaks down the sucrose into glucose and fructose in the honey's stomach, and then they spit it out, and the house bees consume it and add more enzymes, and they spit it out, and so they keep adding more enzymes and in and out, regurgitating the nectar, and so they'll do that over about 20 minutes, and breaking down the sugars. And when the nectar is down to about 20% water, so the whole time they're actually consuming the water that is in the nectar, and when it's down to about 20% water, it's deposited on the honeycomb, and the bees fan it and speed up evaporation to further condense it, and then they stop when the water concentration is down to about 17 to 18%, and finally move it to its final storage location. So bee spit and evaporation is what honey is. So this is sugar was the first sweetener used by man. And I said they extract the nectar and store it in hives. So its final composition, approximately, it can vary depending on the bees and where they're at in the season, and what flowers they are getting the nectar from. But approximately 40% fructose, 30% glucose, 7% maltose, and then some other sugars and minerals and phytochemicals are in there. So this number doesn't add up quite to 100 for that reason. And then about 15 to 20% water is what it is composed of. And you can see from this image, the flowers the honey is derived from has a big effect on what the honey looks like. So like on the left here, we have honey from orange blossoms, and on the right, honey from buckwheat. Okay, oh, one more. Isomalt. So isomalt, this is kind of a weird one because it's kind of weird. So it's a mixture of 1 part mannitol, which is a sugar alcohol, 1 part sorbitol, which is a sugar alcohol. Notice the "-ol." If you see an "-ol" at the end of a chemical name, that means it is an alcohol. Alcohol, all. And then 2 parts beet sugar, so sucrose. Its special properties, it liquefies at 154 degrees Celsius, which is a higher temperature than regular sugar. It colors easily and remains clear so you heat it to a really high temperature without it burning or caramelizing, so turning brown. And sorry, but it liquefies at a lower temperature, but you can heat it to a higher temperature without it. Sorry, back up. This liquefies at a higher temperature than sugar, which sugar liquefies at a lower temperature. So you can heat this up and have still nice, pretty sugar candy compared to pure sugar. Okay, and so these are just a few little examples of things that have been made with isomalt, like this ball here and this flower and like these little fake stones. And it comes in a powder. It can be blown also, so into like little threads of sugar, isomalt sugar. And that is it for this week. So next week we will cover more functionality of sugar as well as nutrition.